Can you hear me and see the slides?
Yes, we can. Looks great.
Thank you very much, Noel, for the extremely kind introduction. And it's a great pleasure for me to speak to you at your Amyloid Day. So I'm going to present the interim data from the first in-human trial of an in vivo CRISPR-Cas9 based gene editing therapy, NTLA 2.01, designed to edit the TTR gene in patients with transthyretin amyloidosis. And I think it's worth saying that this is the first time that any human has been given gene editing in vivo gene editing therapy. These are my disclosures and the aims and objectives. By the end of this session, I hope that you will be able to understand the principle of gene editing technology, understand the role of gene editing for treatment of ATTR amyloidosis, and lastly, understand the potential of gene editing in medicine generally. So the principle of treatment in all types of amyloidosis is to reduce the rate of ongoing amyloid formation, the aim being to achieve a new equilibrium in which the natural rate of amyloid clearance, shown here, exceeds the rate of amyloid formation in order to achieve net amyloid regression. So reducing the rate of amyloid formation may be achieved in one of two ways. The most tried and tested method in amyloidosis generally is to reduce the concentration of the circulating amyloid fibril precursor protein. Or specifically in ATTR amyloidosis, one can stabilize the TTR tetramer in the bloodstream with drugs such as tafamidis and acoramidis. So in ATTR amyloidosis, the approved RNA therapeutics or interference therapeutics, patisiran, inotersen, and vutrisiran, often referred to as gene silencers, work by reducing the circulating concentration of TTR. So here are some images showing amyloid regression in AL and AA amyloidosis. The two panels on the left here show serial anterior whole body SAP scans. So SAP scintigraphy is a quantitative nuclear medicine technique that was developed in our center by Professor Philip Hawkins and is used to diagnose and monitor amyloidosis. So in the absence of disease-modifying treatment, we only ever see amyloid deposits accumulate by this technique. So the two scans in the panel on the left here are taken from a patient with AL amyloidosis who received chemotherapy and achieved a complete hematologic response, resulting in removal of the amyloid-forming light chain from the circulation. And you can see regression of amyloid from the liver, the spleen, and the bones. Two scans in the center panel here are from a patient with AA amyloidosis whose inflammatory condition was successfully treated, resulting in normalization of circulating SAA protein concentration. Again, you see regression of amyloid from the liver and spleen. Now, please note the different rates of regression from different organs and the time between these pairs of scans. Amyloid regression is slow, but nonetheless, it's unequivocally associated with improvement in amyloidotic organ function and prolonged patient survival. And one of the problems with SAP scintigraphy is it's unable to image amyloid deposits in the heart. And more recently, I think you heard from Mariana Fontana, who has been using novel cardiac magnetic resonance imaging techniques to show that amyloid can regress from the heart. We've seen exactly the same thing. In other words, amyloid regresses from the heart in some patients who achieve a complete response, complete hematologic response in cardiac AL amyloidosis. And that is associated with significant functional improvement and unequivocally associated with improved survival. So this slide here demonstrates the importance of the magnitude of knockdown of the relevant fibril precursor protein in relation to clinical outcomes in different types of amyloidosis. In AL amyloidosis, outcomes are significantly better in patients who achieve a hematologic CR. That's equivalent to 100% knockdown of the circulating amyloid-forming light chain, shown by the blue line at the top there, compared to patients who achieve what hematologists call a VGPR, very good partial response, equivalent to about 90% knockdown of the amyloid-forming light chain. And those patients, in turn, do better than those who achieve a partial response, defined as between 50 and 90%, who in turn do better than patients who achieve no response, defined as less than 50% reduction of the fibril precursor protein. The panel in the middle shows the relative risk of death in relation to octiles of serum amyloid A protein concentration in patients diagnosed with AA amyloidosis. So it's notable that the relative risk of death is nearly 4 times higher, you can see here, in patients with a median SAA concentration of between 4 and 9 milligrams per liter, compared to those with a median SAA concentration of less than 4 milligrams per liter. In other words, a normal SAA concentration. And you can see the very clear relationship between the magnitude of SAA knockdown and outcome. Similar data emerging in ATTR amyloidosis, although it's early days and the data, to be honest, is insufficiently mature to be conclusive. However, the panel on the right shows the relationship between circulating TTR concentration after treatment with patisiran and change from baseline in the mNIS+7 neuropathy score. So existing gene silencing therapies such as patisiran, vutrisiran, and inotersen, as I mentioned earlier, reduce serum TTR concentration by approximately 80%. And they've been shown to stabilize and in some patients even improve neuropathy scores in hereditary ATTR amyloidosis. Patisiran has also been shown to reduce cardiovascular-related hospitalizations and all-cause mortality in patients with hereditary ATTR amyloidosis who have cardiac involvement, and more recently, also to benefit patients with predominant amyloid cardiomyopathy. However, some patients, particularly those with advanced disease, continue to experience debilitating effects and die from their disease. Furthermore, gene silencing therapies require chronic and lifelong administration. On the basis of all types of amyloidosis treated so far, as I've explained, one would expect greater TTR knockdown to facilitate clinically meaningful symptomatic improvements accompanied by better clinical outcomes. So editing of the TTR gene, which has the potential to provide permanent and profound TTR knockdown after a single infusion, is a particularly attractive treatment strategy. So in 2012, Jennifer Doudna and Emmanuel Charpentier showed how CRISPR, a naturally occurring biological defense mechanism in bacteria, could be used as a sort of cut and paste tool for editing gene sequences, work for which they received the Nobel Prize in Chemistry in 2020. NTLA-2001 is an investigational CRISPR-Cas9 based in vivo gene editing therapy, and it consists of a single guide RNA molecule that targets the human TTR gene and a human optimized mRNA sequence of strep pyogenes Cas9 protein encapsulated in this lipid nanoparticle, or LNP. Importantly, the therapeutic platform is actually modular. It's extremely easy to make a guide RNA molecule which targets any gene in the body. And that's an important point when we want to consider the potential of CRISPR-Cas9 gene editing therapy in medicine as a whole. So following intravenous administration of the LNP, it's transported directly to the liver, where it's taken up by the LDL receptor on hepatocytes. After endocytosis and release into the cytoplasm, the Cas9 mRNA is translated, producing the Cas9 enzyme, which then interacts with the single guide RNA to form what is known as the CRISPR-Cas9 ribonucleoprotein complex, or RNP complex. The RNP complex enters the nucleus, and a target-specific 20 nucleotide sequence at the 5' end of the guide RNA binds to the DNA double helix at the target site. And that leads to precise cleavage in the targeted TTR gene sequence known as a double-strand break. And then endogenous DNA repair results in insertions or deletions of bases in the target site. And these frame shift alterations reduce functional target gene mRNA and consequently production of the target protein. The plot here shows the in vivo therapeutic index with effects of concentration of the guide RNA on the percentage TTR gene editing by NTLA-2001 in cultured primary human hepatocytes. The vertical line here marks the concentration at which we routinely observe 90% reduction in TTR protein production, known as the EC90. Above the EC90, NTLA-2001 demonstrated saturating levels of TTR gene editing accompanied by greater than 95% reduction in TTR protein. Now, an important question that the scientific team from Intellia asked before bringing this agent to clinic is what is the level of off-target editing at these concentrations? The team performed extensive genome-wide screening with computational and laboratory-based techniques, both cell-free and cellular assays, to detect and validate the likeliest sites for potential off-target editing. And these methods identified 7 sites, importantly all in non-coding regions of the genome. And the experiment shown here compares the rate of on-target and off-target editing following treatment with NTLA-2001. And what you see down here marked by these colored lines, each representing one of the 7 sites, is that there was no detectable evidence of off-target editing, even at concentrations of NTLA-2001 3-fold greater than the EC90. So preclinical in vivo studies were conducted in cynomolgus monkeys using a surrogate LNP incorporating a single guide RNA, obviously specific to the monkey TTR gene. This graph here shows the mean percentage reduction from baseline in serum TTR protein concentration in monkeys receiving the LNP at 0, 1.5, 3, and 6 milligram per kilogram of the RNA cargo on day 0, and followed up for 367 days. The vertical lines represent standard deviations across the 3 animals in each group. Note a single dose at 3 milligram per kilogram achieved a durable reduction in circulating TTR protein concentration of approximately 95% after day 28. Safety studies established a dose range at which no adverse effects of NTLA-2001 were observed, with near complete sustained reduction in serum TTR following this single administration. At higher doses, the adverse effects in the monkeys were dose-dependent, transient, reversible, and monitorable. So the first in-human study with NTLA-2001 is a 2-part open-label, multi-center study initially designed to treat patients with hereditary ATTR amyloidosis and polyneuropathy. The study design was later amended to include patients with predominant ATTR amyloid cardiomyopathy, including patients with wild-type ATTR amyloidosis. Part 1 is a single ascending dose design with a minimum of 3 patients in each of up to 4 dose escalation cohorts, 0.1 milligram per kilogram, 0.3 milligram per kilogram, 0.7 milligram per kilogram, and 1.0 milligram per kilogram. The recommended dose selected in part 1 will be administered to a dose expansion cohort in part 2. And obviously the primary objective of this first in-human study are to evaluate safety, tolerability, PK and PD, the latter by serial measurement of TTR levels. So I will report data on the first 15 patients with hereditary ATTR amyloidosis and polyneuropathy from the dose escalation cohort included in part 1. And bear in mind that all patients received a one-time infusion of NTLA-2001 on day 1 of the study. So here are the demographics of those first 15 patients. I won't read through it. And here are the baseline disease characteristics of those first 15 patients. And patients with 7 different pathogenic TTR variants were represented. And it's noteworthy that most of these hereditary amyloidosis patients had early stage disease. In other words, most had a PND or polyneuropathy disability score of 1 and were in NYHA functional class 1, despite imaging evidence of cardiac involvement in a proportion. And the relatively minor cardiac involvement or complete absence of cardiac involvement in some patients is obviously supported by the fact that NT-proBNP was below 600 nanograms per liter in all patients, as you can see. So NTLA-2001 was generally well tolerated in the acute phase, with the majority of AEs being grade 1, i.e. mild in severity, and mostly unrelated to treatment. All patients received a complete study dose of NTLA-2001, and there were a number of infusion-related reactions, but they were all considered mild and did not require dose interruption. All of the infusion-related reactions resolved without clinical sequelae. There was a single serious adverse event, or SAE, which was that of intractable vomiting in a patient who was known to have very significant pre-existing amyloid-related gastroparesis, and he received a 1 mg per kilogram dose of NTLA-2001. In other words, the highest of the 4 doses. There were no significant abnormalities of liver function or coagulopathy, and all the laboratory excursions were considered clinically insignificant. So here you see the list of AEs. As you can see, the most common AEs were mild and transient headache and mild infusion-related reactions, usually manifesting as fever or chills. As previously mentioned, all patients completed the NTLA-2001 infusion. And here's the so-called surrogate efficacy data. So at day 28, you see dose-dependent reductions in serum TTR concentration. And the mean percentage reduction in circulating TTR concentration from baseline in the 6 patients who received the 1.0 milligram per kilogram dose of NTLA-2001 was 93%. Looking at the 6 patients who received the 1 milligram per kilogram dose in a bit more detail, you can see that all achieved a percentage reduction from baseline in serum TTR of greater than 85%, and that 3 of the 6 patients achieved percentage reductions of greater than 95%. In other words, there were no significant or no major outliers, if you like, and TTR knockdown in this dose cohort was consistent. The reductions in serum TTR reached a nadir by day 28 and were sustained through to month 2, as you can see on this graph. And the reductions in serum TTR among those receiving therapeutic doses of NTLA-2001 was sustained through to the last follow-up, although we do need to acknowledge that the length of follow-up in the higher dose cohorts remains relatively short at the current time. So over 30 patients have now been dosed across the polyneuropathy and cardiomyopathy arms of the phase 1 study, and the growing body of data has demonstrated that at therapeutically relevant doses, treatment with NTLA-2001 results in rapid, deep, sustained, and consistent reductions of serum TTR. There's a plan to evaluate a fixed dose in the dose expansion cohort. So lastly, I think that we, in considering the potential future applications of CRISPR, let's step back one stage, if you like, and think, why is it that ATTR amyloidosis was chosen as the prototypic clinical indication for the Alnylam, Ionis, and indeed the Intellia pipeline programs? Well, there's an unequivocal link between the protein and the disease. You need protein deposition to cause amyloid, if you like. Very importantly, circulating TTR is exclusively liver-derived, and that enables potential successful delivery to the cells of choice, in other words, hepatocytes. On the basis of the earlier data that I showed you, knockdown of TTR was felt to be clinically beneficial, and that has indeed proven to be the case. Lastly, given the overlap in physiological functions of TTR, knockdown of TTR was thought likely to be safe, which similarly has also been proven to be the case. Now, future potential CRISPR technologies and clinical applications include other knockout editing. I've shown you the first example of knocking out a gene via editing. And, you know, there are other programs in the current pipeline, including hereditary angioedema, alpha-1 antitrypsin deficiency, you know, which are diseases amenable to knockout editing. There is knock-in editing, where you can potentially knock in a gene sequence, and that might be applicable, for example, to hemophilia. There's base editing, in which one can edit single bases, and you know that might be applicable, for example, to sickle cell disease. There's something called prime editing, which is editing of single bases or indeed longer stretches of DNA. And also multiplex editing, editing of multiple genes in one nanoparticle, if you like, or one drug. And that might be applicable in the future. We're not there yet, but that might be applicable to certain cancers. And lastly, there's CRISPR activation and CRISPR inhibition, known as CRISPR-A and CRISPR-I, which have the potential to turn genes on and off. So I'd like to acknowledge the patients, the contract research organizations involved in the trial that I presented, and my co-investigators at the National Amyloidosis Center, Richmond Pharmacology, and the University of Auckland, and of course the many members of Intellia and Regeneron Pharmaceuticals who are responsible for the development of anti-ATTR1,